Yes, sir.
हाँ now हाँ screen share तो है ही है, हाँ. Yes. So today we have to discuss about the structure and working of eye. So eye is basically a device, optical device through which we visualize the signal. You know? We visualize the signal, and then according to the information received through the light to the brain, there will be action. So we have to come in this way. This is the visible light. It's just a part of electromagnetic spectrum. So electromagnetic spectrum means you see the wavelength in terms of nanometer. So in electromagnetic spectrum, gamma, X-ray, ultraviolet, then visible. A small range is visible. Then infrared, then radar, television, and then others. So this ultraviolet is above, this is the 400 nanometer, or you can say 390 nanometer to 750 nanometer in the red. So this region is visible, and it is visible because our eye is sensitive. Now the question is, why I can receive only this signal? Why I cannot receive X-ray or radar? So the reason that today we will find it. So this one is cross section of the human eye. You know, this human eye, this part is this outer part. There are three layers of the eye. Basically, first is outer layer, and this outer layer is sclera. This outer layer, white layer, is sclera. This sclera has again three layers. That is outermost, middle, and innermost. But here we will not concern with that sclera. And then there is a middle layer, and this middle layer is another kind of protective layers. And then the inner part is the most important, which is termed as retina. This is termed as the retina layer. Now, before retina, you see the outer side is iris. Outer side is iris. That is the part we visualize. We visualize two parts in our eyes. First is iris, other is pupil, opening of iris, that is pupil. So iris and pupils are two parts that are visualized here in outside, or in inside. In inside there is the another combinations. These combinations are something you call this part is conjunctiva merged with inside of the eye, and in this eye from the out to in there is a lens, which is the most important part of our eye. This lens is made of the material which has more elastic, and the due to elasticity, the focus of length can be changed. So through this elasticity, lens accommodate its focal length, and it will increase or decrease as per the position of the object. As per the position means when the object is near to the eye and when object is far away from the eye. So in the both cases, there is need to change the focus. So lens accommodate the focus. And these are the ciliary fibers that will hold the lens in contact with the main part. Now you see this green and red. This green and red are the blood nerves, and the inner part of this layer, which is retina, that is connected with the uh, optic nerves, which send the information to the brain. One important thing is vitreous humor. vitreous humor. Vitreous humor is the semi-liquid crystal material that is inside the eye, which is in the liquid form of this case. So you see there are most important is two part. One is retina, and other is the important things that is here is lens. Lens and retina, these are the two important and most commonly known parts in the eye. So after the structure, this is used to be asked in examination. Please draw the human eye and level the different parts and explain its working. So these are the human eyes. Now the parts and their working is corona. Cornea. Cornea is transparent outward covering of eye that admits the light, and then pupil, adjustable opening in the iris that regulates the amount of light that enter the eye. If you see carefully, then you find the change of the size of pupil when the object is near and in dark, or in low light position. Then you see pupil diameter is larger, bigger. And when you see in outside, that is in sun ray, then the size of the diameter of pupil is small, so that will allow less amount of light because of the brightness. So the adjustable opening in the iris that regulates the amount of light that enter the eye. Now iris, the pigmented ring of muscles situated behind the cornea, and in this way that we have study about the different part. So since eye is supposed to be optical instrument, biological optical instrument, so we have to concern with the refractive index. So you see the refractive index of eye air is 1. So the refractive index of cornea, that is 1.376. Aqueous humor, the refractive index is 1.336, and then bifocal lens, it is 1.4, and vitreous humor is 1.337. So you see there are two humor. One is aqueous, other is vitreous. Aqueous is outer part, which has the 1.336, and vitreous is inner part, that is 1.337. This is in comparison of the lens. So you find before lens, the medium is rarer, 1.336. A slight different, that is on the third place of the decimal, 336 and 337. So after the passing through the lens, the vitreous humor is have more diffracting index, that is denser medium. So you see cornea has more refractive index, so it is the denser medium in comparison to the air. So in comparison to air, there is a denser medium, and from denser medium to rarer medium, and from rarer medium to again denser medium, that is focal length. And from this focal length, high refractive index to low refractive index, means light, the path of light is from denser media to rarer medium. So you see from this very important concept, the light is passing through the several medium which refractive index are slightly different. So denser to rare, rare to denser, and something like this light will pass through the eye in the human body. You see, the inner part is, that is retina, the neural tissue, and photoreceptive cells. Photoreceptive cells, what is the photoreceptive? Basically, this cell is a transducer are the devices which transfer electromagnetic wave into electrochemical potential. That means it generates the potential. Potential means action potential, and that is sodium and potassium ions, that is electrical charges, which flow from eye to the brain. That is in the form of the signal. That signal is given. So located on the inner surface of the posterior portion of eye. Posterior portion of eye means, yeah, this part is. This is the posterior. That is the optic nerves. So in this optic nerves, these are the photoreceptive cells. And the tissue of neural tissue. Neural tissue means these tissue connects the information from eye to the brain. Now, during the photoreceptive cells, there are two important structure. One is rod, other is cone. So this rod and cones are sensitive towards the color. Color means frequency, which kind of the frequency is there. That is photoreceptor cells of the retina sensitive to the light of low intensity. Cone, photoreceptor cells of the retina maximally sensitive to one of three different wavelength of light and hence encode color vision. So you see the coding and decoding, that is digital electronics, which already inbuilt in our eye in the terms of rod cone on structure. We will see the clear structure of rod cone just after a few minutes. The other part is lens, consists of series of transparent onion-like layers. You see, series of transparent and onion-like layers. Its shapes can be changed by contraction of ciliary muscle. Okay, these are the ciliary muscles which are responsible to change the focal length. That is, that process is known as accommodation. That is accommodation. What is accommodation? You see, changes in the thickness of the lens, accomplishing by the ciliary muscles that focus image of near or distant objects on the retina. So on the retina, in order to make a clear focus that is adjustable, it is through the thickness of the lens, and this thickness of the lens create the variation in focal length, so that light will focus on the retina. So how this is focused, you see in other diagram. Suppose this is the object, and this through this object the light is passing in this way on the lens, and then here it is the retina, and on this retina this image is formed. And this part is the lens, which lens is adjustable through the ligaments. Adjustable through the ligaments. This is the iris, pupil, which allow to flow. You see the cross section. This is the cross section of the pupil. Sometimes it is called aperture, aperture, but here it is said pupil. And then there is a cornea, which cornea is visible, and cornea has a different pigment. Sometimes you see the greenish eye, bluish eyes, whitish eye. It is all due to the cornea color pigments. And ciliary muscles, which protect the lens at a fixed place. Then sclera, that sclera is the white of the outside, which is the outer layer to protect this whole device through the body. And this one is optic nerves that sends the signal to the brain through this optic nerves. So this fovea, area of retina that mediate the most acute vision, contains only color sensitive cones and rods as it is. Next is optic disc, location of retina where fibers of ganglion cells exist, the eye responsible for the blind spot. Blind spot is this blind spot. So this is the back of that, and that is optic disc in this way. So in this way, there is a cross-section of the eyes, and on the basis of the cross-section of eyes, how this eye color transfer to the brain. So this mechanism is as here it is the light incident on the retina, and then in this retina there is the, you see these are the pigmented retina. This is the pigmented retina. This pigmented retina is connected with the structure. This structure is cylindrical. This structure is conical, triangular. So cylindrical, conical, cylindrical, conical, cylindrical, cylindrical. So this cylindrical structure is known as rod cells. Cylindrical structure is known as rod cells. And this triangular structure is known as cones. So these are rod and cones. We will have the difference, compare rod and cones. It is used to ask in the examinations. But you see here it is the, you see number of the rods are more than the number of cones, you see in this slide. So whatever the structure is, first this take the information from pigmented retina and transfer to the bipolar structure. You see these are connecting, these are the connecting points, that is the frequency or wavelength taken from the pigment to transfer the bipolar. And this is the process, in which the electromagnetic energy is converted into electrochemical energy. So what is the I? This is just transfer the energy from this electromagnetic wave to electrochemical wave, and this ganglion part is transfer the action potential, that's the ions to the optic nerves, and this optic nerves transfer the information to the brain, So this working of retina is very important. Here it is the first part, that is the pigmented retina, and this pigmented retina take the information to this. These are the transducers. These are the transfer the energy of electromagnetic energy to electrochemical energy through this bipolar medium, and then it is transferred to the brain through the active potential. Now, contains photoreceptor cells, rod and cone, and associated interneuron and sensory neurons. The detailed diagram, that is again in order to make a clear concept about the vision, rods and cone and their structure, the clear picture as from the natural way. You see this one is the eye, here it is the retina, this small part is retina, and the more detailed diagram of retina is these are the pigmented cells, and these pigmented cells are connected to the cone and rod and cones. So this structure you can see here, it is this structure. These are the cone, his rod, cone, rod, cone, and rod. And this structure is again, you see this is the detailed structure, that is bigger structure, and this bigger structure is here, it is cone, and this is the rod. The rod type of internal structure is this one, and these are the, why this structure is important for us, because you have to understand how the electromagnetic energy is transferred to the electrochemical energy. So the neural circuit of the retina of a primate, the incoming light reaches the photoreceptor cells, that is rod and cones only, after passing through several thin transparent layers of other neurons. The pigment epithelium absorbs light that is not absorbed by the photoreceptor cells, and thus minimize the reflection of the stellite. The ganglion cells communicate to the thalamus by sending action potential draw to the axons. However, the photoreceptor cells and their part is neuron communicated by graded synaptic potential that are conducted. Graded synaptic potential. So again, this one is a colorful, and the diagram which is used to explain the actual part is something like this. You see the actual part. Here it is the disc, that is, this is the rod type of disc. So scanning electron micrograph of the retinal rod cell. So scanning the microscope, which is very, very high resolving powers used to take this kind of the photograph in the eye, that is of the red cell. So what kind of things you are observing here? It is the disc-like structure inside the cell. These structure transfer the electromagnetic energy into the electrochemical energy. So this is the disc, and this is the outer pigment segments, and these are the process through which the whole things. So it is the internal structure. Why it is important? Because you have to understand about what kind of mechanisms used to draw this part. Now you see the difference. These are the important questions. That is rod's outer segment is rod shaped, as we have shown. This one is a rod shape, this one is a rod shape, this one is a rod shape. Here it is the cone shape. So this cone is given. The outer segment is cone shaped. It is in the cone form. Now you see, I said rod shape, number of rods is more than number of cones. So it is 10 to the power 9 cells per eye. Here it is 10 to the power 6 cells per eye, distributed throughout the retina. So use the peripheral visions. Here, this is mainly in the fovea, so can only detect image in center of retina. Good sensitivity. Rod has good sensitivity because of the number is large. Poor sensitivity, it is the lesser number. Only one type, monochromatic vision. This is the 3 type, RGB, red, blue color, that is fundamental colors. We will see how to mix the fundamental colors to give the other type of the color. Many rods connected to one bipolar cell, and this one is here. Each cones is connected to one polar cell. This connection is shown here. You see, this one is a rod, and here it is the rod. These 2 rods are connected. Many rods are connected to this one. While in cone, this one cone is connected to this one bipolar part. This cone is connected. So the cone is singly connected, while the rods are multiple connectors. So this one is because the number is 10 to the power 9 in rod, and number is 10 to the power 6 in cones. Now, this diagram is important for us, being in the students of physics, unless until we have not have any diagram of the voltage potential. So it seems that we are not doing just through our discussion. So light hyperpolarize the plasma membrane of a retinal rod cell. This is the membrane potential. So membrane potential is, you see, minus 30 millivolt. Here it is minus 35 millivolt. So minus 35 to minus 30 millivolt. So light pulse is moving in this way, and this one is the resting potential. The time in second, 1 second. So from 1 second, minus 35 millivolt, 30 to 35 millivolt, and then this kind of the curve is. So if you see this is inharmonic curve. Inharmonic curve means the time taken from minus 30 to minus 35 is lesser than the time taken to minus 35 to 30. It is more. So that's why it is enharmonics. The light induced hyperpolarization is transmitted by the plasma membrane from the outer segment to the synaptic body. Synaptic body is the part of neurons, that is to the brain part, optic nerves. A single photon closes hundreds of catons specific channel. Specific channel, how much is 500, and leads to hyperpolarization of about 1 to 5 millivolt. This 1 to 5 millivolt is known as action potential. These are the photoreceptors, bipolar and ganglion. These are the rod cone shade. This is the again a diagram that is taken from the um, high resolving microscope, and on the basis of that high resolving microscope, again we have the photoreceptors. These are the cones, these are the rods. So rod cones are the structure. These are the connectivity. Bipolar layers are connected in this way. And here ganglion cell. These ganglion cells, what the ganglion cells is doing, this kind of the curves they are forming. So these curves are obtained through this mechanism. So this mechanism is important. So major cell type of the retina which are transferring the energy from this one. Again, this one is the receptive field of the ganglion cell. Ganglion cell is the way to transfer the light. You see, this one is the light, receive light. Here it is the time taken. So these diagrams classify the how much time taken to transfer the light from the retina to the nerves. Now, as I have said, rod is sensitive for monochromatic, while cone is sensitive for mixing of the color. So mixing of the color R, B, Z, R red, B blue, and G, this is green. So red, blue, green. This is the red, blue, green. R, B, G, red, blue, green. These are the fundamental colors which mix all the color segments in the pigment. So these are the sensitive through the cone and other way. You see, another way to express in terms of the wavelength and the relative absorption. So it is the blue cone, and here it is the rod. This is the rod sensitive, that is 4966. And rod, this one is the red cone, this is the green cone, and this is the blue cone. So cone has three colors, that is RBG, this is R, this is B, this is G, and these are the rod cones. The rod is sensitive 496, while the cone is sensitive to 559, 531, and 419 nanometer. So these are the structure which gives the information regarding the case. Now, one problem is color blindness. This color blindness is nothing but the genes for the green and rod pigments lie adjacent on the human X chromosome, are 98 identicals in nucleotide sequence, including intro and UTR. Therefore, are susceptible to unequal homologous recombination. Third, 5% of male have this form of blindness. So blindness kya? There is a green and red pigments like adjacent. What must be there? Green and this red. And here it is the green and red light adjustment in the chrome so that it cannot be detected. And here it is the way how to, this one is the receptor inactive, here the receptor down the regulations, and this the receptor sequence size. This is for the molecular part to be discussed. Now, for the previous classes, you have seen the eye defects. These eye defects are myopia, hypermyopia, astigmatism, and presbyopia. And this another is not defect, but is important for us. This is the persistence of the image. So eye defect is myopia. What is the myopia? Near or short-sightedness, means before the retina. Before the retina, the information is this focus, light focus before the retina. So you have to try to give the focal length increase, and that's why perimedial, the concave lens are used. Hypermyopia, that is far-sightedness, that is the behind the retina. And if the image is behind the retina, then you need to focus the light on the retina. So you want converging lens, and that is convex lens. So myopia people has concave lens, hypermyopia people needs convex lens. Before and after retina in the blur, that the information, that is the images form in two colors, two positions. So for that point, we need the bifocal lens of cylindrical. See, presbyopia, distant objects are clear, but not objects, but the near objects are blurred. And this blur is, this is the blurry object. So reduction in the power of accommodation, that is lower convex and upper concave. Bifocal, whenever the people use the bifocal, then the upper part is convex lens and the upper part is concave lens and lower part is convex lens. Now the important point is fifth, that is persistence of image.